Professor Patrick Johnston, thank you very much indeed for giving us a few minutes at ECANS TV. Just need an update on your spectacular unit in Belfast. This is, gone. I've seen it going through from through puberty to adolescence, and it's now a fully fledged, tough cancer centre.
Well, Gordon, I think you actually may have seen it at conception.
Possibly did.
Quite apart from puberty.
Possibly did, yes.
Indeed, I think you were probably one of the people who lit the flame. It's gone through a huge development over the last 10 years. We now have a modern comprehensive cancer setup in Belfast, beginning with the clinical centre, which opened in 2005. And was the initial reason why I came to Belfast from the United States to help develop that. And in doing that, put in place a modern radiation oncology and medical oncology setup, but also a strong clinical research culture and educational culture underpinning it. And what's great now is to see over 1,000 patients a year in clinical trials, where we do 3 or 4 phase 1 trials a year. And we also have a very large translational trial portfolio. We also have founded a number of very important international agreements, particularly the NCRI All-Ireland Consortium Agreement, which was initially signed in 1999, but is still doing very, very well, has had over 400 nurses going back and forward between the United States and ourselves. But also other health professionals, medical oncologists, scientists, all able to engage in international research activity and training opportunities. The other very exciting thing is that in 2006 we opened the Centre for Cancer Research and Cell Biology, and that now houses approximately 270 scientists, 30 principal investigators. The major areas of interest are around biomarkers in colorectal cancer, DNA damage repair, radiation biology, and BRCA studies, and the BRCA and DNA damage and repair. And that continues to grow, that whole setup. And as a result of all of that, we were announced as one of the first CRUK centres back in 2008. Which we were delighted by. So I suppose in essence for the cancer patient we now have access to both leading therapies, but also the cancer patient in Northern Ireland has become a partner in the whole clinical development process, which they never were previously. We also have, in terms of clinical practice, the very latest state of the art, both from an imaging as well as from a treatment point of view. And we're continuing to hire people internationally as well as locally in that effort.
I think it's a magnificent achievement, and congratulations on that. You've maintained, however, your own lab interest all along, and
Sure.
I also congratulate you on that, because frequently it's the bureaucracy and the time involved in the admin of such
Sure.
a setup suppresses original research in the lab. DNA repair still on your list?
DNA repair, more so death signalling, and how we got started into the death signalling was that we noted that one of the genes that was being acutely upregulated in response to DNA damaging agents, particularly the fluoropyrimidines and antimetabolites, so those that are damaging DNA indirectly, were actually the death receptors. And we found that an inhibitory protein called FLIP, or the Fleiss inhibitory protein, which regulates the death discs, appeared to be the key component of this upregulation. And so we've been looking at novel ways of actually trying to target the FLIP protein to try and knock it out. One of the most recent things that we have found is that there is, it binds to one of the HDACs, and it seems to be regulated particularly by HDAC6, and is then targeted for degradation as a result of this. So we are looking at that interaction site currently in relationship to trying to target this protein. It also, of course, is an exciting novel mechanism around HDAC activity, and one that has not been previously noted. The other area that we've exploited very significantly over the course of the last 4 years has been around the EGFR receptor. Again, we published in Cancer Research 3 years ago that the activation of the EGFR receptor was a critical determinant of whether an EGFR inhibitor was likely to be synergistic with agents like irinotecan or 5-FU. We've gone on to show that actually some of the shedases, particularly an enzyme called ADAM17, is a key regulator of the ligands that cause this activation. And as a result, we are currently piloting an ADAM17 antibody in conjunction with Cambridge in relationship to trying to knock it out, with the idea that this may be a better way of actually targeting growth pathways as a mechanism of acute resistance.
Well, both pathways, you mean the HDAC and the
HDAC and FLIP, exactly.
Okay. Tell me about ADAM.
So ADAM17 it's one of the members of what are called the shedases. The shedases are a group of proteolytic enzymes that are actually transported out of the cell, and what they do is they actually cleave ligands or prepare ligands for stimulation of both death signaling pathways but also growth signaling pathways. So all of the EGFR members, IGF, and so we have found that the level of ADAM17 goes up by 1,000-fold when you treat a cell acutely with chemotherapy. And it is driving signaling pathways for growth. And so therefore we've now, in conjunction with Cambridge, actually begun to look at the role of targeting ADAM17 with a humanized antibody. And our initial in vivo experiments actually look very, very promising.
Is it detectable in the blood?
So yes, we've also developed an ELISA assay for assaying it in blood, and we're actually doing that study at the present time. So we're assessing both its prognostic and predictive capability, and hopefully we will have that data by mid-2011.
All in colorectal cancer? Or
this is all in colorectal cancer, but the same principle. We've actually tested this in lung, in some of the lung models that we have, and we see exactly the same thing. There's a different atom involved. So there's a level of specificity related to the given tissue, I believe, in terms of what shedase is functionally important. So ADAM10 would also appear to be another very important shedase in regulating the creation of these ligands.
And what do the HDAC inhibitors do to this?
So the HDAC inhibitors, in relationship to the FLIP molecule, what they do is they regulate an acetylation event that prevents, when FLIP is acetylated, it actually is prevented from actually being targeted for degradation. When it is deacetylated,
so it
suddenly, exactly, and therefore by targeting it, you actually will decrease the levels of it within cells, and therefore cells will be primed for death. Because the inhibitor, at least from the external apoptotic mechanism, DR4, DR5 receptor generated, the inhibitor will be absent within the cell.
Okay. And the ADAM17 work is going towards a clinical trial.
Yes.
With Cambridge. What sort of molecule is this? Humanized
so this is
antibody?
A humanized antibody, GSK molecule. As I say, we're in the in vivo stage of that, but exactly that, with the context being that we will drive towards clinical development over the 2011-2012. This is the work of Dr. Sandra Van Chaybroek, who's a CRUK fellow in my lab.
Good. Paddy, thanks very much indeed for bringing us up to date. I really appreciate it, and once again, fantastic job.
Thanks, Gordon. Thanks for the opportunity.